And today we shall be breaking the steps down for better comprehension of what happens to produce energy from the carbon-carbon bonds in carbohydrates. The first step in the glycolytic pathway will be the addition of a phosphate group to glucose at carbon 6. So this is your glucose molecule, one glucose molecule. ATP comes in to donate a phosphate group at carbon 6. 1, 2, 3, 4, 5, 6. So at carbon 6, we see the addition of a phosphate group donated by ATP, which becomes ADP. So glucose is converted to glucose 6-phosphate. That's the first step. In the second step of glycolysis, what happens there is simply the isomerization of glucose 6-phosphate to fructose 6-phosphate. If you remember previously from one of our classes when we were talking about isomerizing a carbohydrate, we saw that fructose and glucose are structural isomers. So we see a change from glucose 6-phosphate to fructose 6-phosphate. And so we have glucose 6 becoming fructose 6. You see the change in structure. This is an aldose sugar and this is a keto sugar. So you can see 1, 2, 3, 4, 5, 6. So this carbon is still this. There's a change in the form because this is a keto sugar, it's an aldose sugar. At this step, no energy molecule is involved, just a change in structure to give fructose 6-phosphate. Now the third step of the glycolytic pathway is the addition of another phosphate group to fructose 6-phosphate at carbon 1. So right here at this carbon 1, which is now here, we have ATP again coming in, as we saw in step 1, to donate a phosphate group to fructose 6-phosphate at carbon 1 to give this. So we now see fructose 6-phosphate becomes fructose 1,6-bis-phosphate, 2 carbon, 1 at 6, and now another one introduced at carbon 1. So fructose 6-phosphate becomes fructose 1,6-bis-phosphate, catalyzed by the enzyme phosphofructokinase. And then we go to step 4, we see the cleavage of fructose 1,6-bisphosphate to 2 3-carbon sugars. Now this fructose 1,6-bisphosphate that we have here is a 6-carbon sugar. Most times students struggle with how does this become this. So I included this straight chain form of this compound to show you exactly how 2 3-carbon compounds come out of this when it's cleaved. Just a cleavage between carbon 3 and carbon 4. There's a split here to give us the top, which is the dihydroxyacetone phosphate, and the down, which is glyceraldehyde 3-phosphate. So this is split into 2, a reversible reaction that gives us the top part from here to here. This hydrogen comes here to give this dihydroxyacetone phosphate. And then the lower part of it, where this now becomes an aldehyde group, because hydrogen has been given to this carbon, becomes this glyceraldehyde 3-phosphate. So at step 4, there's a splitting, a cleavage of fructose 1,6-bisphosphate, which is this to show you how it's split to give these 2 3-carbon compounds or molecules. And this is catalyzed by the enzyme aldolase, catalyzes the split here. And then at step 5, what we just have here, though is a reversible reaction, but in the case of glycolysis, the reaction favors, the direction is favored this way. So at step 5, the dihydroxyacetone phosphate changes its form from this to this, is a isomerization. So this is converted to this to continue with the glycolytic pathway, because what will continue this step to step 6 is this, not this. So whatever is converted is gotten from here is tautomerized to give glyceraldehyde 3-phosphate. Step 6 of the glycolytic pathway is the dehydrogenation of the glyceride 3-phosphate from step 5 at C1. So this is glyceride 3-phosphate, carbon 1. This hydrogen is removed by NAD. And then its position is taken over by a phosphate group from inorganic phosphate. So we remove this and we add a phosphate group there. And because the hydrogen is removed, it becomes NADH. And then the hydrogen from inorganic phosphate is released as free hydrogen. Remember, step 5, glyceride 3-phosphate is there, and then dihydroxy acetone phosphate is tautomerized to G3P. So we have 2 molecules from step 6. Whatever you have as substrate now or products, 2 molecules. So 2 molecules, so we have 2 NAD plus 2 phosphate to give us 2 NADH plus 2 hydrogen ion. I've explained. This hydrogen is the one from here, and this one is the one from the free inorganic phosphate that came to donate itself, replacing the removed hydrogen to give us 1,3-bis phosphoglycerate. And this is catalyzed by the enzyme glyceride 3-phosphate dehydrogenase. Step 7, the next step in the glycolytic pathway, we have the removal of one phosphate group from C1, that's carbon 1, to phosphorylate ADP to ATP, step 7. So we'll see this hydrogen, this phosphate group here, is used to convert ADP to ATP. So because we have 2 of this here, each donates a phosphate group and becomes 3-phosphoglycerate. So the phosphate group as carbon 1 is lost to give us 3-phosphoglycerate. And then at step 8 of the glycolytic pathway, what happens there is the mutase converts 3-phosphoglycerate to 2-phosphoglycerate. So we have this 3-phosphoglycerate, 2 molecules of it here. There's a mutual rotation converting it from 3-phosphoglycerate to 2-phosphoglycerate. Look at this very well. What happens is simply a change in the position of the phosphate group. The phosphate group moves from carbon 3 to carbon 2. And the hydrogen is replaced here, and then the phosphate group is replaced here. So there's a swapping in the position of hydrogen and phosphate. Hydrogen comes down, phosphate comes up. So 3-phosphoglycerate becomes 2-phosphoglycerate. And the enzyme here for this reversible reaction is phosphoglycerate mutase. And then step 9 of the glycolytic pathway, what is happening there? Simply the removal of a water molecule from 2-phosphoglycerate. Each of these, remember there are 2 molecules of 2-phosphoglycerate. Water is removed. Water is hydrogen and hydroxyl. So these and these are removed to give us phosphoenol pyruvate. Because there are 2 molecules here, 2 molecules of water will be removed to give us 2 molecules of phosphoenol glycerol. So what is happening here is simply the removal of this hydrogen here and this hydroxyl group here. And then we now see this double bond here. No longer a single bond, but a double bond here. And so we have phosphoenol pyruvate. And then the last step of the glycolytic pathway, step 10, is simply the removal of the remaining phosphate group in phosphoenol pyruvate at carbon 2 is used to phosphorylate ADP to ATP. Remember, it's 2 molecules. So each of it donates its phosphate group as carbon 2 to ADP to give us ATP. And then we have our final product, pyruvate. So 1 molecule of glucose from the beginning of glycolysis gives us 2 molecules of pyruvate. Remember, the enzyme here for this is phosphoglycerate kinase, and the enzyme for this is pyruvate kinase. Glycolysis is so important because it helps in the breakdown of carbohydrates, not just to release energy, which you have seen here. 2 molecules of ATP are invested at the beginning of this pathway at reaction 1, step 1 and step 3. While if you look at step 7 and step 10, has produced 4 molecules of ATP. So, which means that the overall gain of ATP in this whole 10 process is just 2 molecules of ATP. It's produced by the cell each time it undergoes glycolysis. Glycolysis does not only give us ATP. Most of these intermediates you are seeing here find their way to other metabolic activities as well as biosynthetic pathways. I trust that we'll have a nice time looking at glycolysis, trying to demystify the glycolytic pathway, which is very essential for cells in the metabolism of carbohydrates and in the production of various intermediates that are critical to the maintenance of a stable internal environment that can sustain life. Thank you so much for your time. Thank you for learning with us today. Until I see you next time for another exciting tutorial on biochemistry, it is bye-bye for now.